There is a hospital room in Sydney where, for the first time in human history, doctors did something that was supposed to be impossible. They did not cut a single strand of DNA. They did not slice, splice, or rewrite a single letter of the genetic code. And yet, inside the body of a child born with a disease that has killed and crippled human beings for thousands of years, a gene that had been silent since before that child was born woke up. Just like that. No incision. No edit. A switch, flipped in the dark, inside the very molecule that makes you who you are. And when scientists tried to explain how it worked, they realized they had just settled an argument that had divided biology for over 40 years. This is not science fiction. This happened in January of 2026 in a lab at the University of New South Wales, and it is only the first thread in a story that is about to unravel everything we thought we understood about heredity, identity, and the rules that supposedly govern life itself. To understand why this moment matters, you have to go back 70 years to a small room in Cambridge, England, where two young scientists, one 24, the other 36, stayed up night after night trying to solve a puzzle that had haunted biology since a Swiss chemist named Friedrich Miescher first isolated a strange phosphorus-rich substance from the pus-soaked bandages of wounded soldiers back in 1869. Miescher called it nuclein. He had no idea he was holding the blueprint of every living thing on Earth. It would take almost a century before James Watson and Francis Crick finally cracked the shape of that blueprint, the double helix, in February of 1953. A discovery so foundational that it became not just a scientific milestone, but a cultural symbol printed on museum walls and etched into the public imagination as the moment humanity finally saw its own instruction manual. But here's the part most documentaries leave out. Watson and Crick did not just discover structure. They discovered a set of rules, a hierarchy. DNA makes RNA, RNA makes protein, and protein makes you, for 70s. Years that one-way arrow was treated almost like scripture. Genes were the blueprint, and everything else, how you were raised, what you ate, what your grandmother lived through, was supposedly just noise around the edges. Then, in 2026, within the span of a few months, 3 separate discoveries detonated that assumption from 3. Different directions at once. And the strange thing is, almost nobody outside a handful of laboratories noticed it happening. The first crack appeared in a place nobody expected to find it, in a pond. In May of 2026, researchers using a new single-cell DNA sequencing method were running what should have been a routine experiment, cataloging the genetic material of microscopic organisms living in ordinary pond water. And then something didn't fit. A routine experiment with a new single-cell DNA sequencing method turned into a surprising scientific twist when researchers stumbled upon a bizarre genetic code in a microscopic pond organism. This was not a mutation, it was not a disease, it was a single-celled creature, a protist, quietly living its entire existence by breaking one of the supposed universal laws of molecular biology. The very rules that every textbook, every classroom, every scientist on Earth had been taught to treat as unshakable. Instead of following the near-universal rules of life, this newly identified organism rewrites how genes signal their end. Think about what that means for a moment. Every cell in your body, every cell in every plant, animal, and bacterium ever studied uses what scientists call the genetic code. A near-universal language where specific sequences tell a cell's machinery precisely where a gene's instructions stop. It is one of the few things in biology considered close to sacred, a rule so consistent across billions of years of evolution that finding an exception felt. To the researchers who found it, like finding a country where gravity pulls sideways. And this organism had been living right under our noses the entire time, in ponds, doing something we assumed was biologically forbidden. The scientists at the Earlham Institute who made the discovery didn't go looking for it. They stumbled into it by accident. That accident alone should terrify and excite you in equal measure. Because if one of biology's supposedly universal laws can be quietly broken by a pond organism nobody had bothered to sequence, what does that tell us about how much of the rulebook we have gotten wrong? What else is hiding in the 8 million or so species we still haven't even cataloged? And more importantly, if the rules of the genetic code itself are not as fixed as we believed, what does that mean for the second discovery, the one that struck even closer to home, the one that didn't happen in a pond, but in the genes of mice, and by extension, quite possibly in you? That second discovery came out of a collaboration stretching from Johns Hopkins to Texas A&M to the University of North Carolina, published in Nature Genetics in May of 2026. And it targeted something even more sacred than the genetic code. It targeted Gregor Mendel. If you remember one thing from a high school biology class, it was probably Mendel's P's, the tidy, elegant laws of inheritance that a 19th century monk worked out in a monastery garden, laws so clean and so mathematically satisfying that they became the foundation stone of modern genetics, dominant and recessive, predictable ratios, a closed logical system. And for over a century and a half, those laws held. Then researchers using long-read DNA sequencing technology, a method powerful enough to examine DNA fragments over 1 million base pairs long, discovered something that should not have been possible under Mendel's framework. Scientists found that certain inherited epigenetic marks, chemical modifications that influence gene activity without altering the underlying DNA code, can be transmitted across generations in ways that do not follow Mendel's classic rules. And let that settle for a second. This is not about the DNA sequence itself changing. The letters of the code stay exactly the same. What changes is something layered on top of that code. Invisible chemical tags that tell a gene when to switch on and when to stay silent. And researchers discovered that these tags can be passed down from parent to offspring in patterns that completely defy the neat, predictable math every biology student has been taught for generations. One of the lead researchers, Casper Hansen of Johns Hopkins, put it plainly. This work may convince scientists to integrate both genomics and epigenomics, more often for a complete understanding of how traits that produce disease and healthy states are inherited. Translate that out of academic language, and here is what it actually means. Whatever your grandparents lived through, the famines, the traumas, the diets, the chemical exposures may have left chemical fingerprints that were passed down to you, not through changes in your DNA sequence, but through an inherited layer sitting on top of it, a layer that operates by its own rules, rules we are only now beginning to map. And once you understand that, the story stops being abstract. It becomes personal. Because it means the disease you may be at, risk for, the trait you can't explain, the thing that runs in your family that doctors have always attributed to bad luck, might actually be an inheritance nobody knew how to read until now. Which brings us back to that hospital room in Sydney, and the discovery that ties both of these threads together into something with the power to reshape modern medicine within our lifetimes. For decades, one of the deepest disputes in molecular biology revolved around a question that sounds almost too simple to have caused so much argument. Do the chemical tags sitting on top of our DNA, the ones controlling whether a gene is turned on or off, actually cause genes to be silenced? Or are they just a side effect? A footprint left behind after silencing happens for some other reason? It sounds like a technicality. It is not. Billions of dollars in gene therapy research, and the fates of patients suffering from some of the most brutal inherited diseases on Earth hinged on the answer. In January of 2026, a team at the University of New South Wales finally found it. A new CRISPR breakthrough shows scientists can turn genes back on without cutting DNA by removing chemical tags that act like molecular anchors. The work confirms these tags actively silence genes, settling a long-running scientific debate. For the first time, scientists proved directly and conclusively that these chemical anchors are not passive scenery. They are the lock, and if they are the lock, then instead of cutting the gene itself, the way traditional CRISPR gene editing does, slicing DNA like a molecular scalpel and hoping the repair goes exactly right, scientists can now simply remove the lock. No cutting. No risk of the DNA healing incorrectly. No risk of unintended mutations cascading through a genome that has just been sliced open. Just a quiet, surgical removal of the very thing keeping a gene shut, and the gene switches itself back on, using machinery your body already has. This gentler form of gene editing could offer a safer way to treat sickle cell disease by reactivating a fetal blood gene. Sickle cell disease, a condition that twists red blood cells into rigid, crescent-shaped weapons against the very body that made them, clogging vessels, starving organs of oxygen, causing pain so severe that patients describe it as being crushed from the inside. It affects millions of people worldwide, disproportionately, devastating communities in Africa, the Middle East, and among people of African descent across the globe, communities that have waited generations for a cure that always seemed to remain one breakthrough away. And now, researchers believe they may have found a way to reactivate a fetal gene, one your body used before you were born and then silenced. A gene that, if switched back on, could compensate for the broken adult gene entirely. Researchers say it opens the door to powerful therapies with fewer unintended side effects. But here is where the story stops being a simple tale of triumph and starts becoming something far more complicated, because every single one of these three discoveries points toward the same unsettling truth. The rulebook of life is not what we thought it was. And the moment you can rewrite the rulebook, you open a door that cannot be closed again. Consider what it actually means to possess the ability to switch human genes on and off without cutting DNA, cleanly, safely, at scale. The same technology capable of reactivating a silenced gene to cure sickle cell disease is, in principle, capable of reactivating or silencing almost any gene whose behavior we understand well enough to target. Genes tied to muscle growth, genes tied to pain perception, genes tied to aging, to metabolism, to the production of hormones that shape mood, aggression, even the biological signatures of fear. For every parent desperate to cure a child of a devastating inherited disease, there is somewhere down the line a defense ministry, a pharmaceutical conglomerate, or an ambitious government quietly asking a very different question. What else can this technology touch? History gives us an uncomfortable answer to what happens when powerful new biological tools arrive faster than the ethical frameworks meant to contain them. When the structure of DNA itself was mapped in 1953, nobody in that small Cambridge lab imagined that within a generation, that knowledge would be feeding recombinant DNA technology, then genetic fingerprinting used in courtrooms and criminal investigations, then designer gene therapies, and eventually the CRISPR gene editing revolution that, within the past decade, had already produced the first gene-edited human embryos, an event that triggered global condemnation, prison sentences, and a scientific community forced to reckon in real time with just how far ahead the technology had raced beyond the guardrails meant to govern it. Every leap in our understanding of DNA has been a leap toward power, and every leap toward power has eventually attracted the attention of people who see biology not as medicine, but as strategy. That is not paranoia, it is documented history. State-run biological research programs throughout the 20th century repeatedly blurred the line between defensive science and offensive capability, and intelligence agencies across multiple nations have, for decades, tracked advances in genetic engineering with the same intensity they track breakthroughs in nuclear physics or cyber warfare, because the underlying truth is the same in both cases. A tool capable of healing is, by definition, a tool capable of harming, and the only difference between a gene therapy clinic and a bioweapons lab is intention, funding, and who gets to decide where the line is drawn. When you can flip a genetic switch without leaving a trace of surgical intervention behind, when the very chemical tags controlling human biology can be manipulated with a precision that leaves the underlying DNA sequence completely untouched, you have created something almost undetectable by design. No scar tissue, no obvious foreign DNA insertion for a forensic geneticist to flag. Just a switch, flipped quietly, with consequences that could ripple through not just one person's body, but if the epigenetic inheritance research from Johns Hopkins is correct, through their children and their children's children, in patterns that don't follow Mendel's predictable math at all. For the first time in the history of genetics, we are looking at a technology that could, in theory, alter not just an individual, but the trajectory of a bloodline, invisibly, silently, and without anyone downstream ever fully understanding why they inherited what they inherited. And yet, before the fear takes over completely, it's worth remembering something equally true. The overwhelming majority of the people chasing these discoveries are not chasing power. They are chasing an end to suffering that has existed for as long as human beings have had children. Somewhere right now, there is a watching her son curl into a fetal position on a hospital bed during a sickle cell pain crisis, gripping his hand, unable to do anything except wait for the medication to work and pray that this crisis isn't the one that damages his organs permanently. There is a father whose daughter carries a genetic mutation nobody in the family understood until a long red sequencing test finally explained why certain symptoms had haunted 3 generations of women in his bloodline. Women who were told for decades that their pain was psychological, that their fatigue was laziness, that their conditions were simply unexplainable bad luck. The scientists working in these labs are not comic book villains hunting for control over the human genome. Many of them are driven by exactly the opposite instinct, a desperate, deeply human need to finally answer the question that has haunted families for generations. Why did this happen to us? And can we stop it from happening to the next generation? That is the tension sitting at the heart of this entire story, and it is a tension with no easy resolution. The same long-read sequencing technology capable of uncovering hidden epigenetic inheritance patterns that could finally explain mysterious inherited diseases is the exact technology a state actor would need to map a population's genetic vulnerabilities. The same CRISPR breakthrough capable of curing sickle cell disease without cutting DNA is the exact mechanism a bad actor would want if they were trying to develop biological interventions that leave no genetic scar behind. There's no version of this story where the cure exists without the risk sitting right beside it, holding its hand. Which is exactly why what happens in the next few years matters so profoundly. Because right now, in 2026, we are standing at almost the exact moment Watson and Crick stood at in 1953, holding a blueprint we have only just learned to read, except this time the stakes are exponentially higher. Because we are no longer just observing the AM blueprint. We are learning to edit it, to silence and awaken it, to understand that it is inherited in ways far stranger and more layered than Mendel's P's ever suggested, and to realize, thanks to a humble pond organism nobody expected to matter, that even the supposedly universal rules underlying all of it may have exceptions we haven't found yet. Every week, in labs scattered from Sydney to Baltimore to a research institute in the English countryside, another piece of the puzzle shifts into place, and every single piece raises the same uncomfortable question that has haunted every major scientific leap in human history. From the splitting of the atom to the invention of the internet, not can we do this? We already know we can. The question that actually matters is who decides what we do with it next, and how long the world has before the technology outruns the wisdom needed to control it. Deployed not in a hospital but in a laboratory, with a very different mission statement, one where the goal isn't curing disease but engineering advantage, where the line between medicine and weapon dissolves the moment intention changes, and nobody outside a classified program ever finds out, until the consequences are already inherited by people who never consented to carry them. 70 years ago, two young scientists in Cambridge thought they had found the final secret of life, the shape of the molecule that explained heredity once and for all. They were right that they had found something extraordinary. They were wrong that they had found the whole story, because it turns out the double helix was never the finish line. It was the opening chapter of a much longer, much stranger book, one where the rules keep shifting the deeper we read, where Amongst Peas gave us a framework that mice have just shattered, where universal genetic code turned out to have exceptions hiding in water the entire time, and where the very chemical locks controlling human biology have just been proven, for the first time, to be exactly as powerful as scientists always feared and hoped they might be. We are not at the end of the genetic story. We are barely past its preface. And the truth this documentary leaves you with is not a comfortable one, but it is an honest one. Humanity now holds a set of tools capable of rewriting the biological inheritance of our species, quietly and visibly across generations, and we are being asked right now, in real time, without the luxury of decades to deliberate, to decide what kind of species we want those tools to build. Because somewhere out there, a child with sickle cell disease is waiting for that switch to be flipped in their favor. And somewhere else, unseen, someone else is waiting to see how far the switch can be pushed before anyone notices it's been touched at all. The blueprint of life has just been rewritten twice in 1 year. The only question left is who gets to hold the pen next.